Okay, good evening, ladies and gentlemen. First of all, I would like to thank the organizing committee for the invitation to present part of my research studies to you. I am very honored by this invitation. Today, I would like to dedicate this talk to Dr. Noemi Waxman for her outstanding contributions in the natural products chemistry, and who I had the pleasure to meet in the past when we were part of the CTED program on natural products for neglected diseases. I am currently a researcher at the Federal University of Rio de Janeiro, where I have founded the Phytochemistry and Pharmacognosy Laboratory in 1996, where we studied Brazilian medicinal plants by the use of ethnopharmacology and random screening approaches. Today, I will talk about a subject that speaks to my heart, which is the story of our research studies over the last 10 years with Amazon medicinal plants, and in particular with the Amazonian medicinal plant Saracura mira. To address this subject, I will go a little back earlier in my career to the year of 2006 and talk about the beginning of this project. We began this work many years ago with the PhD thesis of this guy here, Dr. Danilo Oliveira, who studied the medicinal plants used by Killambola communities of Oriximiná in the north of Brazil, in the Amazon region. In that work, a total of 251 medicinal plants were cited, among them Saracura mira, which is Ampelosisyphus amazonicus, used as a tonic and fortifying agent, as well as to treat and to prevent malaria. But where is Oriximiná, this strange town with a strange name? Here you can see the map of Brazil, with the state of Pará highlighted here in the north of the country. And in this window, the small town of Oriximiná that we can see here, highlighted in this larger window, where we can see also the Killambola communities that are divided into 8 territories, highlighted here in different colors. And who are the Quilombolas? Many of you may have heard the term maroon, which was coined to designate the fugitive black slaves from the West Indies and Guyana in the 17th and 18th centuries, and also a descendant of such a slave. In Brazil, by definition, Quilombola, or also remnants of Quilombos, are the descendants of the Africans who were made slaves in Brazil during the 16th and the 18th centuries to work on the farms. The term Quilombola comes from the Tupi-Guarani language, Canhibó, and means the ones who escapes a lot. It is estimated that today in Brazil there are over 3,000 Quilombola communities, of which at least 300 are located in the state of Pará. They have their right to the land secured by the federal constitution since 1988, and it was in the municipality of Oriximiná that for the first time in 1995, a Quilombola community received the collective title for their lands. The Quilombola people from Oriximiná live far from the urban centers, deep in the forest, and so they live from fishing and hunting, also from the collection of edible fruits. Here in this picture, you can see this guy here preparing the acai berries to be consumed. And they also live from the collection of the Brazil nuts, which is a very, very important commodity for this group of people. They also live from the collection of medicinal plants and preparing the copaíba oil, as well as the andiroba oil. Their knowledge comes, why is it so important to work with the Quilombola knowledge? Well, their knowledge comes from a strong African matrix, as well as from the knowledge from the Portuguese colonizers, as well as the Amerindians, which were the original inhabitants of Brazil. Since they live distant from the urban centers, they are in full contact with nature in a region with a high biodiversity. So that is why their knowledge in medicinal plants is so rich. Their knowledge is retained by what they call the great sacacas, which are the pajés, or also known as shamans. And also their knowledge comes from the knowledge of local specialists, which are people that are considered healers, traditional healers. The plant Ampelosiziphus amazonicus is used in all the Amazon region to prevent and to treat malaria. But it's also used as a fortifying agent, as a tonic, aphrodisiac, stimulant, and depurative. It is one of the top 10 medicinal plants among the Killambola from Oriximiná. The plant is consumed as a necros bitter drink. Here we can see this guy preparing the drink. This is the plant in its habitat. And here he is preparing the drink that is also known as cervejinha or cerveja de índio, that means Indian beer. It is an aqueous bitter drink and which produces abundant foaming when it's being prepared, as we can see here. Despite from the fact that the plant is being used in all the Amazon region to treat and to prevent malaria, as reported here in this paper published by our group with this plant, in vitro tests with the plant extract against Plasmodium falciparum gave negative results, as we can see here in this table published in the paper, showing that there is no activity of the aqueous extract from the bark in Plasmodium blood stage forms. However, the plant has been reported to be effective in controlling the infection induced by sporozoid forms, as reported by other publications, not from our group, but from other groups in Brazil. So our interest in this plant arose because of these apparently controversial results. In this way, we thought that it might be possible to suggest that the plant could act as an antimalarial, but as an adaptogen by enhancing the immune system function that would be responsible to combat the disease, and also the plant could alleviate the inflammatory disorders caused by malaria. In fact, ethnopharmacological studies indicate both stimulatory and energetic properties for Ampelosiziphus amazonicus, which is a characteristic feature of adaptogens. So according to our understanding, there is a strong evidence that points the adaptogenic properties of these plants. The term adaptogen was coined by the Soviet scientist Nikolai Lazarev in 1958, and it can be briefly defined as compounds that increase the ability of an organism to adapt to environmental factors and to avoid damage from such factors by increasing the non-specific resistance against stressors, improving one's ability to adapt to stress. In fact, there is no doubt that many plants' adaptogens are able to modulate the distinct phases of Celle's general adaptation syndrome, represented here in this figure, by reducing the stress reactions in the so-called alarm phase, where there is an activating of the hypothalamus pituitary adrenal axis, and retarding or eliminating what we call the exhaustion phase. That is when all the body's resources are eventually depleted and the body is unable to keep a normal function. And the results can be the onset of a disease or of an illness. Lately, there has been a growing interest in the market of dietary supplements with adaptogenic properties. So here we can see in this picture, for instance, ashwagandha, which the Indian plant Vitamina somnifera, the Korean ginseng, the Siberian ginseng, also the Peruvian maca, which scientific name is Lepidium mayeni, among so many others. These plant adaptogens have been shown to be of particular interest in sports medicine by improving the physical performance, but also improving cognitive functions, and also demonstrating anti-fatigue effects. So taking into consideration all the aspects presented so far, we engaged in the study of the barks of this plant in order to provide a new ingredient for food applications. So we undertook the study of their immunomodulatory properties, in the study of its chemistry, and also of their biotechnological applications. To accomplish this task, we engaged into a plant collecting expedition called the Malungo Expedition, involving 6 Killambolas and also 5 members of our university. The Federal University of it comes from the Bantu African dialect, which is an African dialect, which means, among other meanings, companion, brother, but also companion in suffering in the Kikongo language. So it was the perfect name for the expedition. River represented here. This is the town of Oriximiná, and this is the Erepeguru River. So we followed up this river in canoes above the water, full of rocks and rapids and waterfalls. So this part of the river is non-navigable to larger boats. This region was originally registered in the maps of this lady here called Madame Otavie Coudreau in the 1900s, when she made an expedition to describe the geography of this region after the death of her husband. Her husband, the French geographer and explorer, Henri Coudreau, and he came to Brazil to map the tributaries of the Trombe River by the expenses of the governor of Pará, shortly after marrying Octavie Coudreau. However, Henri died before he could accomplish his tasks, and he died of malaria, and after Henri's death, Octavie continued the expedition initiated by her husband, and in 1901, she published the book Voyage au Cuminé. So our starting point was the Pancada community, which is the farthermost community of the Erepeguru River. And we started drinking the Saracura drink because of an alleged outbreak of malaria. So we drank this water extract preparation all the days of the expedition. We used canoes. Here are the pictures of the canoes being prepared to reach the land above the waterfalls. Here we can see the team of the expedition in one of the canoes. Here on the right, you can see the rapids and some the rocks under these rapids. And here we can see the engravings, the engraved stones, which are some stones with, I don't know if you can see here in this picture, the engravings here. And it's interesting that these engravings were represented already in the maps of Madame Coudreau. And what is amazing is that the names of the places that we visited and that we followed, they remained, they are still the same. They were kept unchanged all over these years. So was the name of the island, the Monkeys Island, called in Portuguese Macaco Island, here represented in these piece of the map of Madame Coudreau, shown here in this map. And this is the place, this beautiful picture taken here. This is the collection place where we collected the Saracura Mirá. This, in these pictures here, you can see our camping, and that we're sleeping in hammocks, because in the Amazon region, nobody camps on the ground, it's dangerous, so we have to sleep in hammocks. So back to the laboratories, here are the barks that were dried, processed, milled, and affording 16 kilos of bark, which were then processed in an industrial plant in Brazil, medicinal plants processing plant called Centroflora, who prepared an aqueous extract as a single batch for all the biological applications. This water extract was concentrated and then finally dried by a spray dryer in our university in a final process yield of 4.5%. One important determination before all the biological evaluations was the determination of the daily oral dose for animal testing. This determination was based on the traditional method of preparation and posology, which considers the preparation with 1 teaspoon of bark extracted with 1 glass of water. This yields a final quantity of 0.2% of solids. And if we assume that a 70-kilo person drinks this beverage in a daily doses, approximately the person will take like 6 milligrams per kilo. So we decided to work with a slightly higher dose of 10 milligrams per kilo to biological evaluations and also to formulate a drink. So this is the first paper of the group, issue of the group. And here we can see the first results on the immunomodulatory properties of the plants, confirming the hypothesis of the adaptogenic activity. The plant demonstrated induced an in vivo modification of the B lymphocyte response, confirming the alleged hypothesis of the adaptogenic activity. We are also able to demonstrate the anti-inflammatory properties of the extract, which are partly due to a reduction in cell migration in the inflammatory model, in the inflammatory models that we evaluated, and also to the reduction in the production of inflammatory mediators. In this paper here, we can see the results of the immunomodulatory response of the plant's extract. In this paper, we used a malaria infection model to investigate the immune response mediated by the plant. So here in these two graphics, we can see the increase in the total serum antibodies, IgM and IgG, produced after 5 and after 8 days of treatment of Plasmodium chabaudi mice infected with the Plasmodium chabaudi and treated with SM, which is Saracura mira, or the plant extract. So in comparison to the control, in the 8th day, we have a significant increase in the production of IgM as well as in the production of IgG. By this time, we also began to study the chemical composition of these extracts. And here we can see the HPLC-MS profile of these aqueous extracts with two distinct zones. Here, the saponin zone, and here we can see that in these aqueous extracts, the free triterpene aglycons are also extracted, and they elute here in this between 5 to 10 minutes. This analysis also showed that this saponin zone is zoomed here, and this complex saponin profile is characterized by deprotonated molecule ions, negative deprotonated molecule ions in the mass range of 800 to 1000. With these results, a patent was deposited in Brazil in 2014, published later in 2016, and then finally in 2020, this patent was issued to our group. In the next work from the group, we engaged into the investigation of the powder properties of the spray-dried aqueous extract of the plant, which could be easily dried without no encapsulating materials or without any drying adjuvants. This is interesting because when using a spray dryer, some feed solutions can be easy to dry, but others may be very difficult, especially with plant extracts that can generate highly hygroscopic powders, and consequently adjuvants have to be added. Surprisingly, this was not the case. So this powder contains only the concentrated active compounds of the plant and could be applied as an intermediary product for further food preparations. The powder has good rehydration characteristics and also a high saponin content of 33%. In the present study, in this next study, the potential role of the plant as a general immunomodulatory agent was again evaluated, but now this time we dropped the malaria model. We used it, in this case, animals that were not immunized. But first of all, because the plant extract is rich in saponins, and saponins can have effect on blood cells. We evaluated whether a 12-day oral treatment would alter the circulating red blood cell levels in mice, but as we can see here in this first graphic, no alteration was observed, which is a very nice result. So here in this slide, we can see the results of the plant extract on the basal antibody serum concentrations. And at this time, as I said before, we dropped the malaria model using mice in the absence of immunization. The animals were treated with different antigens. We used lipopolysaccharide, LPS, and dextran as antigens, and we recorded the increase of the antibody levels, IgM and IgG, produced by these antigens. So here in this first graphic, we can see that in the animals treated with the plant extract, after 7 and 14 days, there is an increase in the IgM levels when the animals were treated with LPS, and also there is an increase after 14 days in the IgM levels of the animals treated with the plant extract. As for the IgG levels of the animals treated with dextran, there is an increase of the titer of these IgM levels of these antibodies that keeps after day 21, these levels are increased as compared with the control groups. So these were very also very nice results because this natural product shows that the plant has a remarkable effect on increasing the basal level of antibodies, suggesting that the plant is able to boost the natural antibody levels and therefore improve the host resistance. So our observations, these findings, they are in accordance with the description of what is considered a plant adaptogen. There are compounds that increase the non-specific resistance against stressors, improving one's ability to adapt to stress. And also these observations are consistent with the popular use of the extract from the plant as a stimulant, as an energetic, as well as a fortifying agent. So in this same work, we continued the study of the saponins from the plant. But now we engaged into some studies with high performance and high resolution mass spectrometry of this plant extract. This was a collaboration with an Italian group of Dr. Rastrelli in Salerno, in Italy, and also with the collaboration of my sister Gilda Leitão, who is a specialist in countercurrent chromatography. So this research with HPLC high-resolution mass spectrometry revealed that the main saponins present in the extract are composed of three main Damaran aglycone skeletons, group 1 being represented by jujubogenin aglycones, and groups 2 and 3 composed of a 16 keto Damaran saponins, being this third one composed by a C31 16 keto Damaran, with an extra methylene group here, 24 methylene group, as compared here to group 2. And this is a newly proposed skeleton type. The proposal of this C31 aglycone was possible based on the work of Brandão and co-workers, who published in 1992 published the isolation of a new Damaran aglycone skeleton type with 31 carbon atoms named ampelose genine. So apparently this is a feature in the biosynthesis of those triterpene saponins. In this slide, we can see in detail the diversity of the saponins in the bark of this plant, showing at least 19 different ones, bearing 3 to 4 sugar residues, as well as ester residues. And at least, in this study, at least 6 saponins with 31 carbon atoms in the aglycone could be identified. And this is supported by the fact that all these 6 saponins have in common this loss here of 156 units that is related to this alkyl side chain here, bearing an extra methylene group in position 24 as compared to saponins in the group 2. And this alkyl side chain here is lost by a macrolafferty type rearranged as compared to saponins of the group 2. In an attempt to isolate this complex substance from the plant extract, a collaboration with the group of countercurrent chromatography group in Brunel University in the UK was started. And in this work, we report the separation of the saponins of this plant by skeleton type. So in this work, a one-step countercurrent chromatography separation afforded the separation of the oleanan type saponins from the damaran type saponins in this one-step separation. In a further separation of this group, the jujubogenine saponins were separated from the keto-damaran type saponins. The fractions in these separations were analyzed by liquid chromatography coupled to high resolution mass spectrometry, as shown here in these slides, the profiles of each fraction. And by applying this strategy, the number and type of structures identified was expanded to 28 different ones, which are represented here in this table. It's too small. I understand that you cannot see each one, but they were separated and expanded to different ones. At this point of the journey into the discovery of the properties of these plants, one issue that bothered me during this research is the fact that most of the material, the plant material collected, was being tossed away. That is, from 60 kilos of plant material, only 16 kilos of bark were being used. So why use only the barks? Why not use all the plant material? Does the wood contain saponins? And if yes, is their chemical composition similar? This task, to analyze this task, it was entrusted to our PhD student, Simone Mendonça, with the collaboration of Dr. Rosineide Simas, who is a mass spectrometrist especially, under the supervision of my sister, Gilda Leitão, and me. This girl then initiated the study of the chemistry of the woods of this plant in order to identify the saponins in bark and wood by mass spectrometry. The methodology of the study was first to build a custom database of mass spectrometry library with a literature search of the saponins already described for the plant, not only for this plant, but also within the Rhyminaceae family also, which is a family that produces a lot of saponins compounds. To obtain and align the chromatograms obtained for the bark and wood using the MZMine tool, and then combine this data with data from high-resolution mass spectrometry data obtained by electrospray ionization in an Orbitrap analyzer operated in the negative ion mode. The analysis of the fragmentation patterns demonstrated here in figures A and B, but in the article there are more other fragmentation patterns suggested, were also used to identify the saponin aggregates. So with this methodology, Simone tentatively identified 95 different saponins, of which 22 are already had already been described in this plant. 73 saponins were being proposed for the first time to occur in this plant. out of these 73 saponins, 26 were already described for other plant species, and 47 were being proposed for the first time. This work has been recently published in Phytochemical Analysis. It was published online in July 2020, and finally published in print on 2021. Well, last but not least, I would like to present some more recent research, some biological results that I consider that are the cherry of the cake of this talk today, which are a new set of experiments that were designed to evaluate the effects of these plants on the physical performance in rats. The tests were designed to evaluate the maximum speed and time spent on a treadmill with a 10-degree slope. We also evaluated the effects of the plant extract on organ tissues, such as the muscle strength of the soleus muscle, and also the fat percentage of the rat body, among other factors. The preliminary results with the maximum speed tests at the treadmill did not reveal any difference between Saracura and control groups. Here we can see the control and Saracura groups before supplementation with Saracura, and after supplementation with Saracura, showing no effects. That is, the plant has no effect on the physical performance, which could be an action expected from an adaptogen. On the other hand, this is the interesting result. The oral administration of the plant was able to increase the maximum strength developed by the soleus muscle, which is this muscle here represented in the hind paw of the rat. And when we compare the treated group, the Saracura treated group with the control, we can see that there is an increase in the strength of this muscle, which is a very exciting and very interesting result that justifies the future use of these plants extract as a functional food ingredient for athletes training. Another very interesting result from this research is related with fats. When comparing the adipose tissue weights in the animal of the different groups, it can be seen that there is a significant difference, a reduction in the retroperitoneal adipose tissue between the control and Saracura groups. There is a reduction in the treated animals, and there is also a reduction in white fat of these animals, but not in the brown fats. Although these are preliminary results, they are very enthusiastic and promising to the future exploitation of this plant raw material and it could be used, for instance, in athletes to enhance athletes' performance. So, in conclusion, the hypothesis that Saracura mira could act as an adaptogen plant was demonstrated by our group. We also demonstrated by the possible biotechnological applications and the potential of this plant material as a new food ingredient. And also the complex chemistry of these plants begins to be unveiled. It's interesting that the chemistry of the plant has remained practically unstudied for the last 20 years, since the first report of those 3 saponins isolated and described by the group of Brandão in the 90s. But Brandão and her groups and her group were working with the roots of the plants that apparently has a more simpler chemical composition than the bark that contains, as we have demonstrated, circa 90 different saponins. So, which is a very complex chemical composition. So the studies with the plants, these plants are still underway. And finally, this year, at the beginning of this year, in January 2022, the compilation of these results has been published in this review paper from our group in the Plants Journal, emphasizing this plant as a proposed Amazonian adaptogen from these plants. So with this, we hope that we have considerably over this more than 10 years journey, we have contributed to the scientific knowledge of this important Brazilian medicinal plant. And before I finalize this talk, I would like to acknowledge all the teams and the groups that have contributed to this, to the results that I have presented here: the phytochemistry, the food research group, immunology group, the CCC group, our collaborations in Italy, our collaborations in the UK. And also, I would like to thank especially the Killambola group, the people from Oriximiná, who were also always very kind and provided housing for us, not only the Malungo expedition, but all the times that we had to go back to the communities for more plant collections and data collection. And I would also like to say thanks to my university and to the scientific, to the funding scientific agencies, and to my School of Pharmacy. And thank you for your attention. And this talk is also dedicated to the Amazon people. Thank you very much. Gracias por su atención.